Hey guys, so I want to talk about organelles today and a little bit about the different labor division with the cytoskeleton at the end. So just to cover a little bit of ground first, the organelle that we talked about in the previous videos, we talked about a lot of them, but we left some out. So one of them is the lysosome. From the Golgi apparatus, a vesicle can be filled with a lot of enzymes being protein. So the same thing as for the milk, you get to the Golgi, but instead of expulsing that protein out, which you might do in the stomach to digest stuff, you have organelles that are just vesicle full of enzymes ready to digest, let's say, a mitochondrion that is out of function, that is too old. You digest it back and then you release all those parts to make new things or things that come from the outside of the cell, be it debris or more food. And the lysosome will be formed by an invagination of the cell membrane inward, encapsulating those debris, and the lysosomes coming from the Golgi apparatus will merge with that new vesicle with the debris and digest the debris and release the end product for building new stuff. Another type of digestion or like destruction can be done by peroxism. Those are also membrane bound organelles, but those contain a protein crystal that's literally like a mortar, and that's a mortar and pestle, system. So the peroxism protein crystal in it will just crash down. It's also used for toxic waste because enzymes will not function correctly with toxicity, right? Okay, another organelle that we haven't talked about that's specific to plant is the vacuoles. So those are plant cells here, and you can see those green chloroplasts. It's an organelle we'll talk in a minute, but what I want you to focus on is the vacuole, that giant space here. The vacuole simply contains water, and that vacuole helps to create pressure. So the more water is stored in the vacuole, the more pressure is exerted on the outside, on the walls around each of the cells. And we'll talk about those walls. That's the green layer here. We'll talk about those walls later in the video. So that vacuole is specific to plants. Now, in terms of energy, when we separate between animal cells here and plant cells, we produce energy differently, although plant cells have mitochondria as well as animal cells, but only plant cells have chloroplasts. So a little bit of foreshadowing for later in the semester when we learn about respiration, due to like done in the mitochondria. We learn all about the process of respiration from starting in the cytoplasm up to the mitochondria. You need to know as of now the structure of mitochondria. It's a double membrane bonded organelle. There is one outer membrane and an inner membrane that's extremely invaginated. If you remember in our lessons before, the more invagination, the more folds, the more surface area. So mitochondria inner membrane has a lot of surface area. It's extremely important because that's the place at which electron transport chain take place. And those electron transport chains where energy is made. The inner membrane encapsulate comprises the matrix. The matrix is where you find the DNA, the ribosomes, and each of those invagination, each of those folds is called a cristae in the mitochondria. That's what it would look like on an electron microscope with a cut. Of course, it's in 2D. So that mitochondria, those mitochondria are present in animal cells and in plant cells, but in plant cells we have what we call chloroplasts, and those chloroplasts are what allows plants to take energy from sunlight and make sugar. So that's also a double bonded organelle with an outer layer and an inner layer, as you can see here. But inside the matrix of a chloroplast, there are all of those thylakoid stacks that are called, in that the matrix in chloroplast is called stroma. The thylakoids are those little circle discs filled with chlorophyll, sorry. And the stack of thylakoid is called a granum. So the thylakoid stacks into a granum. The grana are found in the stroma, that is the inner part of the chloroplast. There is an inner and outer membrane, and that's the whole chloroplast. We'll talk in detail, that's also foreshadowing, about the photosynthesis and how this works between the light and the and the dark reaction, light dependent and light independent reaction or carbon reaction. We'll talk all about that later on. But the chloroplast is where you can take energy from sunlight to make sugar. Respiration takes place in mitochondria, and it's where you take sugar and you break it down into energy. Both animals and plants do it. Only plants have chloroplast and can take the energy of sunlight. They are self-sufficient, or called also autotroph. Autotroph. We only have mitochondria, so we call heterotroph. Heterotroph, as I spelled it before. Now, the last part of that video is all about the cytoskeleton. Because when you look at those two cells here, they are actually holding together because of something. Those things are floating in the cytoplasm, all those organelles, but they hold together. So now I want you to visualize a room. Why do I choose a room? Because cell means room in Latin. So in a room, that's why a cell in a prison is also called a cell. But a cell in biology is the room in which all the organelles are. So think of that room and think of a cytoskeleton as all poles, ropes, ladders, all of those structures that will help move around in the room, right? You got that in your head, a room with poles, ladder, and ropes? Well, cytoskeletons are made of three major components. The microfilament, that just actin molecules, and actin is a protein. So proteins make everything, right? That's simply beads, like strings of beads attached together. If you go to intermediate filaments, it's like those bridge cables. If you have seen before giant cables, they are torsades or like twists of smaller cables. So if you zoom in, that smaller cable has a little twist, the bigger cable is twist of the smaller cables, and the giant cable is twist of a twist of a twist, right? Russian doll style. The microtubules, it's tubulin subunits, but this time they make a tube, and that tube is extremely important for movement. So let's see what they do. The microfilaments, this is a full cell. You don't see the cell membrane. There's another one here and here, two different cells. There's a nucleus. And all of the red parts here, those are microfilaments. And they really help to contract. So there will be ropes from side to side of your room, and you can tug on it to contract the cell, right? The intermediate filament, they're all about mechanical strength. So think of those as springs. If you contract the cell, what allows it to bounce back is those intermediate filament. If you press your finger against another finger on your hand, you don't leave indent because your cells spring back thanks to the intermediate filament. The microtubule, on the other hand, you can see them here in green, and here during a mitosis event. This is actually anaphase on mitosis. We'll learn all about it later in class as well. We use them as trackways. So think of the microtubules as tubes, as we've seen, and organelles or anything where you can latch onto it and those will climb. There is a full video you can look up online. It's extremely cool. There's a full protein that actually walks on the microtubules and pulls the organelle behind it. It's pretty cool. You can look it up on Google if you are really interested. And that's a microskeleton, your cytoskeleton. I talked earlier in another video about the cilia and flagella, how that works. That's also part of the cytoskeleton. This is a detail of the enzyme that works at the bottom of the flagellum. And so that's really all the really cool thing that cytoskeleton does, but it's not only inside, it's also outside. The plant cells have that cell wall. So remember that cellulose, that's a macromolecule of carbohydrate, lots of sugar. And when you make a mesh of it outside of your cell, you end up having that really rigid structure encapsulating the cell. It's extremely useful, and we learn a lot more about it when we talk about osmosis for plants, and we talk about tonicity and all of that later on. But really, that idea of a room with all the structure in it will really help you to understand how important cytoskeleton to keep things together and move things around in a cell. That's all for today. You guys take care, and I'll see you later.